Hello everyone, I welcome you to my channel Teacher Dulce. In the previous section we have discussed about that the cell is the smallest identity of life. Now, anything beyond cell is not existing. We don't call it as life. Okay, and then we have seen why the cells are small and what is the size of the cell. We have also seen what are the essential features of a cell. Now moving on, before I move on, I would just also like to tell you that shape. I talk about the shape of the cell, and you know that the shape of the cell is related to its what function. And you must have seen different kind of cells, like your nerve cells. It is longer in size, so why is it longer? Because it has to transmit messages. So shape is related to its function. And second important thing just I want to tell you people is that if the size of the organism is big, if the size is big of an organism, does it mean that the cell size is also bigger? No, it is only the number of cells increase, number of cells increase in that case. Okay, that is there, the increase in the number of cells. If I take a rat and a elephant, the number of cells in elephant will increase, not the size of the cell. Okay, this is one thing about that. And now moving on, quickly I will deal with the cell theory. So before going to cell theory, I would like to tell you that Robert Hooke, okay, it was your Robert Hooke, in the year 1655, he observed the Koch cells, which you know that these cells are dead cells under the microscope, and to him they appeared him to be womb-like structures. Okay, and he called them as what? Cells. Cells at that time were very small, womb-like structures. Cells means rooms. Okay, which where prisoners were stored. And then after the Hooke's work, okay, two scientists came in the year, that is, it was Sheridan, he was a botanist, and Schwann, he was a zoologist. Now both of them worked independently, and then both of them worked independently and gave you cell theory. After examining many organisms, they said he all the first point that they said that all organisms are made up of cell and its products. Okay, now the second point they said that all functions are performed by cell and its products. Okay, now uh, just you can see that this theory came quite later. Okay, after the Hooke's work, it came around 1838. Okay, now there was another scientist who added third point to it, and his name is Rudolf Virchow. Okay, his name is Rudolf Rudolf Virchow. He expanded this theory, and the third point was added by him was that cells arise from pre-existing cells. In cells by cell division. Okay, by cell division. Okay, now he said that कि cells cannot be formed de novo, है ना, न्यूली नहीं बन सकते हैं. Okay, और ये हमेशा they come from your which was cells which was existing earlier. Okay, so this was the point which was added by Rudolf Virchow. Now we will move on to our what are the different types of the cell. Okay, now moving to that, we will see what there are two fundamental now types of cells. So let's look at what are the basically your two fundamental mental types of cells. And these two fundamental type of cells are what? Your prokaryotic cell, and second is your eukaryotic cell. Okay, now if you look at these, let's look at what is the difference between the two cells. So by drawing the diagram, I will just show you. Now if you look at a prokaryotic cell, you know, pro means what? Pro means primitive. If I divide this word pro and carry on means, see, pro means before, or you can also say it to be primitive. Okay, that is called as pro or before or primitive. And carry on means what? Nucleus. So it has a primitive type of nucleus. Rather, we don't see nucleus in them. What do we see in them? We see that they have got a DNA. They have got a DNA. Circular DNA is there in them. Okay, and this DNA is located at a region which we call as nucleoid. And no other organelles, you don't find any compartmentalization in the cell. Okay, no compartmentalization. Like, I can give you an analogy. It's a kind of a house which is made up of only a single room. Okay, and in that single room all kind of activities is being done. Okay, and eukaryotic cell, if you look at, it's a different. U means true. U means true. U means U means true. So they have a true nucleus. Now if you look at these cells, these cells are, they look how? See, do they have a nucleus? They have a membrane. Okay, and what else they have? They also have got various other kind of structures in them. Now membranous structures in them. You can see. Okay. Uh, okay, other membranous structures are also you can find in them. So you can find that it's a house with many rooms. So each now rooms have got a different function. Now since you're like, suppose these are the different rooms, suppose these here we call them as what? We call them as organelles. Okay, these rooms are called as organelles. Okay, now like you know about mitochondria, huh? So they have got what? Their role is to produce energy. Now if you look at, both of them have those common structures which I talked in the previous class, that both have got your what? Cell membrane, as you can see. Both of them has got the genetic material. Now inside this nucleus, what is there? There is the your DNA is located. So here your genetic material is compartmentalized. It's separated from the cytoplasm by your what? By your nuclear membrane. So this is your nuclear membrane. So this is your nuclear membrane which is absent here, but you can see the genetic material. You can see the genetic material is present in both of them. Okay, but the nuclear membrane, this one which I am trying to show you, this is absent. This is absent. Okay, in case of your prokaryotic cell. Okay, and here what do you find? You will see that you have made some dots like structures. Okay, now these dots are nothing but your ribosomes. Okay, and remember that these ribosomes, they are present in both the cells. Okay, here also they are, these ribosomes I can make, and these ribosomes are present here also. There is difference in both the kind of ribosomes, but both are ribosomes. Okay, both are your ribosomes. Okay, but there is difference in the two. uh, Now ribosomes on their size is different, and other things are also different. We will talk about it later, but remember that both are ribosomes, but ribosomes are not surrounded by any membrane. Whereas if you look at mitochondria, Golgi body, chloroplast, all of them have got a membranous structure. So they are non-membranous organelle. Okay, so we find that it's a room, it's a house with a single room, and it's a house where we find what? We find it's a house where it has got many rooms. So here is what? Here is your, what is taking place is your division of labor. डिवीजन ऑफ लेबर, है ना? हर एक रूम का अलग अलग कार्य है, isn't it? Now in this case what is happening, a single room is doing all the function. अगर एक ही आदमी को आप सारा काम बोल दीजिए तो definitely his efficiency is going to be less. Whereas if that work is distributed to different people, then what will happen? The efficiency of that person will increase. Okay, of that system will in fact increase, isn't it? So here what do we find, that they, here these kinds of cells, they have got what? There is division of labor and they have got better efficiency. They have got better efficiency. So with better efficiency they can work. Okay, so this is what the fundamentally I've discussed what is the difference between a prokaryotic cell and a eukaryotic cell. And remember, your prokaryotic ribosome is of 70S type. It is of 70S type. Just you remember it right now. I will tell you what is 70S, what does S stands for. And here in case of your eukaryotic, the ribosome is of what type? It is of 80S type. It is of 80S type. Just remember at this moment, we will, as we will move on, it will be unfolded to you what is 70S and what is the difference between them also. Okay, now moving on, I will just give you a basic idea about I will show you a bacterial cell and what other structures also a bacterial cell has apart from these structures. Okay, I will just draw a bacterial cell for you. Okay, this is your bacteria. Okay, bacterial cell. I'm just trying to draw it for you people. On the one side I will show you the structures which are found on in all the bacterial cell. Okay, so you can see that it has got a cell membrane and a cell wall which is present in all the bacteria. There is another structure which I am making. This is called as capsular layer, which is found in some of them. Not all the bacteria has got this capsular layer. And now you can see that I'm just drawing for you. This is your flagella, which is also, it's not that all the bacteria must be having it. And just this is your DNA. Okay, circular DNA they have, which is present in all the cells. Okay, it's present in all the cells of a bacterial cells in all the, and now these are your ribosomes which is present in all the bacterial cells. Now let's see, I will make something else also. Now they have got what? Plasmid. Now you can see this is your plasmid. I'm just labeling it plasmid. Okay, what is plasmid? Plasmid is an extra chromosomal DNA. Extra chromosomal. Look at the word extra. ये extra है. Apart from the main chromosome, which you can see here, this is your main chromosome. So apart from this main chromosome, this plasmid is there. Therefore, we are calling it as what? Extra chromosomal. We are calling it as extra chromosomal DNA. Okay, and it gives certain properties to the bacteria. We will talk about it later. Now it has also got um, certain enfoldings are there, okay, on the membrane, which you don't find in other cases, okay, like I'm trying to locate it. Okay, like this, they have got this, the membrane gets folded inside, okay, and these are your, you can say that they are your what, they are membrane enfoldings, brain enfoldings and foldings. Enfoldings which now these membrane, depending upon their function, we call them as thylakoids if they do photosynthesis, we call them as mesosomes in respiration if they also take part in cell division. So these membrane enfoldings are having different roles, okay. Now these are your, as you know, mm, these dots are your ribosomes, and again I'm repeating for you that these ribosomes are of 70s type in bacterial cell. Now apart from this structure, they also have got, uh, you can see this structure which is, we call them as pili, okay, which has got role in what, in their sexual reproduction, okay. So we will see about sexual reproduction, then we will see there, and they have got also a thread-like structure which is bigger than this, okay, which we call as fimbriae. We call it as what? We call it as fimbriae, hmm, which is used for attachment for to a surface, okay, to a surface. So they help in attachment to a surface, and the pili is there, which is playing role in sexual reproduction. So you can see here, this flagella, this is your flagella, or you can say flagellum as a singular is there. So this is your flagellum. Now this helps in movement, okay. You know that its role is in movement, and I will also tell you how this bacterial flagellum is different from a eukaryotic flagellum, okay. They are different, basically of what they are made up of. And if you look at this, now they, this is their what, this is their membrane, this is your cell membrane, okay, and this is what, this is your cell wall, and you know their cell wall is made up of a special substance which is called as peptidoglycan, which is made up of sugar and attached to a polypeptide, okay. So we will see about it when we will deal with biomolecules, okay. So this is something, in very general I have told you what are the things which is present in almost all the bacteria. They are your cell wall, cell membrane, again I'm repeating for you, and your uh, genetic material, that is your DNA and ribosomes, and definitely this is your what is this, this is your cytoplasm, it goes without saying, plasm, okay. Now these structures are commonly present in all the bacteria, and the structures which are additional, present in some, it may be present in some and may not be present in some, are your flagella, fimbriae, pili, your plasmid, your membrane enfoldings. These are the structures which are may be present in some and may not be present in some. Now, in the next slide, I will show you what is the difference between a gram positive and a gram negative bacteria, which you have most commonly heard about it. Therefore, I'm just telling you the difference between a gram positive and a gram negative bacteria. Now, if you see this one is your gram negative. Now, from where this came, gram positive and gram negative? What is gram? Hans Christian Gram, okay, was a scientist, okay. He was a scientist who said, what he did, he used crystal violet, okay. He used crystal violet used crystal violet to stain these bacteria. He used this crystal violet to stain this bacteria. To stain this bacteria, and if the bacteria retains on rinsing it, then they rinse it. Rinsing means washing it with alcohol. So on washing it with alcohol, if it retains their stain, then it is called as what? It is called as gram-positive bacteria. And if you look at the structure of this gram-positive bacteria, what do you find here? That they have got a thicker layer of peptidoglycan. This layer is what? This is your peptidoglycan layer. Whereas if you look at this, now peptidoglycan layer is very thin in gram-negative bacteria. And in gram-negative bacteria, there is an additional feature. You can see there is a membranous covering. Like here, there is a membranous covering. Similarly, they have a membranous covering here as well. Okay, so they have got this gram-negative bacteria has got what? This is your membrane and a thin peptidoglycan layer. Okay, and here in case of gram positive, they have got a thick peptidoglycan layer. Okay, so this membrane doesn't allows the stain to stain this peptidoglycan, and here there is no membrane, therefore it gets stained. Okay, that's why, and clinically it's very important. Clinically, for microbiologists, the staining, because the staining gives different properties to the bacteria. Okay, so clinically they are very important for them. So I have tried to discuss important things about prokaryotic cell to you people. In the next class, we will start with eukaryotic cell. Thank you. Bye bye. God bless you.